Hi and welcome to my poster presentation. My name is Kümmerle and I will talk about Probeset selection for targeted spatial transcriptomics. In single-cell transcriptomics, you measure the RNA level of all the genes in your cell within single cells, so for thousands of cells. And with this RNA levels, you have profiles for each cell that gives you the information which cell type the cell is, or also in which state the cell is. A very emerging field is spatial transcriptomics, where you get additionally the spatial information to your genetic information. There are methods that are limited in the number of genes that you can measure. For example, you measure 100 genes. And the goal of this project was to select a set of n genes for such experiments. So our starting point was to say, okay, we have the single-cell RNA-seq data set, and we find all this variation that describes the states, and we would like to have a gene set that best captures this overall variation. We've built up an evaluation suite that evaluates a selected gene set, and we have as a first metric this general variability recovery metrics. Then as a second objective, we measure how well are we able to distinguish cell types, or classify them. Then we also don't want to select genes that give exactly the same information, which are totally correlated. So we also measure how the general correlation within a gene, the gene set. And then to select such a set of genes, we use feature selection methods. And for these methods, we also can compute actually the computation time. All right, so we took a lot of very basic feature selection methods. We have a PCA loadings-based selection, differentially expressed genes, sparse PCA encodings-based selection, as reference, highest expressed genes and random genes, and also highly variable genes. And what we found is that PCA loadings lead to the highest performance in our variability recovering metrics group, whereas differentially expressed genes best perform in cell type classification. And we also see that actually the worst performing random reference is best in the redundance group, so that all the genes are completely uncorrelated. But it seems that there's rather a sweet spot in correlation to reach the best performances. Based on these findings from our evaluations, we built a Probeset selection pipeline that optimizes for the variability recovery, but also the cell type classification. In the first step, we have a PCA loadings-based selection. We take those genes, train forests for cell type classification, and additionally we train forest on genes that are selected by differential expression, and then we compare these two forests and check which genes from the differentially expressed genes do we need to add to improve performance of our PCA forests. Additionally, you can incorporate prior knowledge in the selection pipeline. Prior knowledge can be technical constraints, so if your genes are too highly expressed, they will saturate your microscopy image. If they're too lowly expressed, you won't have any signal. You can also provide a pre-selected set of genes and then combinatorially select around the pre-selection, or set a constraint like a minimum number of markers per cell type. And then the pipeline aggregates to a final probe set with also some information why which gene was chosen. Now we compared such a probe set that was selected by our pipeline to a literature-derived set of genes, and also to PCA and differential expression. And what we see is that the probe set really performs very well on the variability recovery and also cell type classification, and has also a lower redundance compared to the other methods. And we also see that the literature-derived gene set doesn't perform so well here, for example, in the cell type recovery, and could also be better in the classification. And so you can, with the evaluation suite, you can evaluate your probe set and check how can you improve it. So in a conclusion, we've built up this evaluation suite, which allows you to benchmark feature selection methods and also to evaluate a given probe set and identify how to improve it. Then we've built a probe set selection pipeline that optimizes for multiple objectives at the same time while including prior knowledge. Thanks a lot for your attention, and thanks also to everyone who was involved in the project. So we are from the TICE group. Special thanks to Malte Lücken, who has the supervisor role in this project. Thanks to Lukas, with whom I built the code package for the user that he can use the method at the end. And this was the approach was together with the DiscoWare consortium. We got a lot of input, first of all from Kerstin Meyer's group, the data sets, a lot of information, and then from Christoph Samakovli's group, all the information about experimental constraints. Thanks for your attention.